Welcome to chapter 20. I am redoing this recording, so if you're in my summer 2026 course, I want to let you know that basically the changes I made will not be reflected on the exam that you're taking in summer 2026. These changes will be reflected rather in the fall of 2026. So if you are taking me in fall of 2026, please note that these changes are going to, can be reflected on your exam. But I wanted to update anyway because this is a lecture I did some new work on. And since we're going to be dealing with antibiotics in this chapter, I chose Alexander Fleming, who discovered penicillin, to put as my artwork. And I chose a quote from him himself: "The unprepared mind cannot see the outstretched hand of opportunity." If you guys kind of know, he went on vacation for about 2 weeks and had a contaminated petri dish that had mold growing in it. When he came back, he discovered that around that petri dish, that bacteria was not growing, and he was like, something's going on here. They investigated, and eventually, after some time, they were able to turn this into something as treatment for as an antibiotic. The first antibiotic, penicillin, was born, and it was the so-called magic bullet that I'm going to talk about. So here are the objectives we'll be talking about. There's some things I added, and we're going to get started. Really, when we talk about antimicrobials, whether we're talking about antibiotics for bacteria, antivirals, antiparasitics, antihelminthics, we can refer to this whole world as chemotherapy. And chemotherapy isn't just like, hey, I'm treating cancer. It is also that I am going in and I am treating a disease, cancer, bacteria, virus, whatever. We can go back to Paul Ehrlich in the 20th century, where he made salvarsan, which was an arsenic-based compound that they used to treat syphilis, Treponema pallidum, which was an STI that was rampant. So basically what this chemotherapy, the antimicrobials, what they do to some degree is they mess with something about a microbe. They're going to mess with things like cell wall or cell metabolism or genetic expression, structure and integrity of the cell wall, that, cell, stuff like that, cell membrane. They're going to do something to mess with the microbe, okay, in the host. Now, an antibiotic, what is really cool about them and what really blows my mind about them and what I really kind of get really excited about is they are made by bacteria to fight other bacteria. It is really a weapon made by a microbe that they use to outcompete other microbes. Because when it comes to competition within the same species is far more intense than competition between members of different species. Because if I'm the same species, we have the same needs. If we look at antibiotics, actually there's over half, more than half, is made by a group of microorganisms called streptomyces. Now, very important, antibiotics are going to do jack squat for a virus. Viruses are inherently not alive in the respect that they're only alive when they're infecting you. And they are not going to have a physiology that we can interrupt. What we have to do with antivirals is do things like prevent the virus from infecting or binding. We can do some things to prevent replication. There's some things we can do, but they're not alive. So if you have a viral infection, antibiotics are not going to do a thing. You might as well sprinkle fairy dust on it. It's just not going to do a thing for that. If it is a microbial infection that is bacterial, then you have to get the right antibiotic. And why is that? Is that there is kind of this idea that not every antibiotic works on every microbe. And we'll talk about that. Now, one thing I want to talk about is called selective toxicity. And what that is, guys, let's say we had a weapon, okay? And this weapon could only harm our enemy, okay? That it would not hurt our allies. That if we fired it and it was friendly fire, our ally would be unhurt, but the bad guy would be stopped, okay? That's what selective toxicity is. It kills the pathogen. Now, I don't like to really call them pathogenic bacteria. More and more, I like to call them opportunistic bacteria. You see, a bacteria is just fine in the right place. E. coli is just fine in your colon, not fine in your urethra. It depends on where it is. So selective toxicity leaves my cells alone, doesn't harm me. It harms the microbe. So we say it's selectively toxic, not to my cells. So we call it a magic bullet for that reason. It affects the microbe, leaves me alone. Okay? Doesn't kill my cells, kills bacterial cells. All right. So penicillin, as I said, was an accidental. And that's really fascinating. Is Fleming goes on vacation about 2 weeks. He comes back, and his petri dish had some fungus growing on it, a colony of a fungus called penicillium. And he noticed that around the penicillium, the bacteria didn't grow. And they were inhibited. They were stopped from growing, and it was interrupting them. And he's like, oh, what's going on? This was 1928. It was 1940 before we actually got it in clinical trial. Now, given that, sometimes I like to tell that story. So my father was born in 1944, and he is the youngest of 13. Alive. He's not the youngest that was born. He's the third from being the youngest, but he's the youngest living of the 13. The two oldest died, and they got strep throat, and they could not get penicillin because it didn't exist. They were born before 1940. And they died from that. They died from strep throat. They got rheumatic heart and died. And they were adults. So we get it basically from the UK to the US, World War II. By World War II, we have this to help treat soldiers and treat people. And it was miraculous to a degree. It saved people, it saved so many lives. But it can't treat every bacteria. We're going to talk about that. So as I said, most antibiotics come from streptomyces. And you can see a lot of these streptomyces, okay? We have, now we have like things like Bacillus subtilis, where we get bacitracin. There was a patient named Tracy, who it had a bacillus from a patient named Tracy. Polymyxma, where we get polymyxin. A lot of these streptomyces, we get a lot of these things like chloramphenicol, these kinds of things, chloramphenicol. We get a lot of these things. Neomycin, streptomycin, gentamicin, some of these come from different things. So there's just many different species, but a lot of them come from streptomyces. What it is these are microbes who are making things that is trying to fight other things. So a fungus might get attacked by some other organism, and I want to put them down so that I can thrive. I want to outcompete them. I want to beat them for resources. These are weapons made by microbes to actually fight diseases. Okay. Now, let's talk about antibiotic spectrum. Very important. Now, when I say it's very important, is the idea is that an antibiotic doesn't affect every bacteria out there that exists. So you're in the hospital and you got an infection. And one of the things you want to know when you do this is what species are they infected with? What are they infected with? Even to the point sometimes what strain of what species are they infected with? And this is going to determine the treatment. Now, what they might do is they might come in with an antibiotic that is extremely broad spectrum, because getting something in them and getting them started is a way to start off. Like, okay, we're going to do this, and then we're going to figure out what it is, and then we're going to attack it with a very narrow spectrum. So what is that? Antibiotic spectrum is, okay, this microorganism can be killed or inhibited by these antibiotics, or this antibiotic. So this antibiotic can affect these microorganisms, okay? All right. Now, so I come in, I got definite signs of bacterial infection, and I'm in bad shape. And if they don't treat me, because in the time it takes them to figure out what I have, I might die. So they're going to start out giving me a broad spectrum antibiotic. They're not giving me something like gosh, I'm trying to remember the species here. Meropenem. There we go. My brain couldn't make a meropenem. Meropenem is like the freaking most broad antibiotic there is. And what we would do is we would get, okay, here's the bacterial isolated. We've identified it. We now know this is what it is. Now we can use this antibiotic or this cocktail of antibiotics to treat it more specifically, more narrow. So let's say you have a narrow spectrum. It might only affect gram positives, may not affect gram negatives, if any. So it's very limited. Narrow spectrum of activity, limited. Broad spectrum, I can handle a bunch. Bacteria are very good to kill with antibiotics. If you have, what we do, if we say an antibiotic is bactericidal, it kills them. Okay, it's going to make them pop. It's going to make them die. It's going to do something to interrupt crucial, I can't live without stuff. Bacteriostatic just says I can't grow. And then this allows me to fight it and use other treatments. We can use a bacteriostatic, like something like a sulfa drug or something. Can be a lot of those are bacteriostatic. And we can either let treatments of other treatments kind of work. Okay, so here's a diagram that I use for antibiotic spectrum. You do not have to memorize this. What I'm showing you is here we have some bacteria. Like we have the different kinds, like we have MRSA and MISA. MRSA, methicillin-resistant Staphylococcus aureus, methicillin-sensitive Staphylococcus aureus, and a Streptococcus, okay? And you can see, okay, penicillin G is just going to take care of Streptococcus, like strep throat, okay? Now, I can get in here to, some of these other to be able to get that. But I'm not going to be able to get MRSA. If I'm going to get MRSA, I need clindamycin, doxycycline, vancomycin, the TMP, SNX to affect metabolism. And we'll talk about some of these, not all of them. So you'll see that, hey, getting some of these, like, hey, these like penicillins, mostly worthless over a lot of your gram negatives. Not very good there, okay? Sometimes they're very good. Sometimes they're extremely broad, okay? There's some really extremely broad antibiotics that we can give to people, and there's some that are extremely narrow, and they only work on this very small set. And we need to know, okay, do we give doxycycline? Okay, what would we do to tell us that is we would do some things like, okay, in infectious disease, what species are we dealing with? We do sensitivity studies and figure out what can I attack them with. Okay, all right, so I wanted to show that. Now, basically, what do antibiotics do? So this shows me, okay, this gives me an idea of what I mean by the antimicrobial spectrum. So what is it that these antibiotics that we've been talking about, what do they do? Well, there's a list of 6 kind of big ways they're going to fight a bacteria. One of these can be inhibiting cell wall synthesis. Basically, like your penicillins, what they do is they interrupt the synthesis and ends up weakening peptidoglycan. What we basically have is this thing called transpeptidation and transglycosylation. that are processes that happen to make sure the cell wall is made. So the cell wall gets weak. And when it gets weak, the cell pops or lyses. We can inhibit protein synthesis. Now, if you think about it, if I inhibit protein synthesis, the bacterial cell doesn't have needed proteins to live, to do essential cell functions. So that will kill it. One of the ways that we do this is affect the ribosome. The ribosomes of bacteria are 70S, where the ribosomes of eukaryotes are 80S. And the fact that the 70S and 80S ribosomes are subtly different, we have selective toxicity for 70S ribosomes. And we can affect only bacterial ribosomes. We can inhibit nucleic acid synthesis by various ways. We can mess up enzymes that are needed to produce it, or a variety of things. We'll talk about that. We might essential nutrients be denied in some cases for bacteria. We can interfere with DNA replication. This is how some of these things like things like Cipro and stuff like that work. Or transcription. We can go in and mess up transcription of mRNA from DNA. We can mess up DNA replication, like go in, take our topoisomerases or gyrase and inhibit it, and the DNA shreds itself. We can injure the cell membrane. That's what some of these things like basically we weaken it, and basically you put holes in it, you injure it. The cell gets leaky, it loses things, it loses metabolites, you mess with the sterols, things like that. We can also inhibit metabolites. Like we can say, hey, you can't get folic acid all of a sudden. And now you can't make essential nutrients that you need for the cell, and it dies because it doesn't have a nutrient it has to have. Okay, so we'll talk about how these things work. So, as I said, you can go in here and we can affect the cell wall. A lot of these things that affect cell walls are psyllins, sporins. Now, bacitracin, you find that in the topical antibiotic creams, like that you put on cuts, like the cream, Vanc, things like that. We can inhibit protein synthesis, some of your other mycins, your tetracyclines, your cyclins, chlorophenicals, and all that. Synthesis of essential nutrients. We talk about the things like T, MP, and SMX. And polymyxin damages your cell, injures your cell membrane. Inhibit replication, your fluoro and chloroquinolones, rifampin, these kinds of drugs. We'll talk about how they do that. If you can prevent DNA from being made in RNA, you can't make proteins and the cell loses things it needs. We can mess up that replication of DNA with like your quinolones, fluoroquinolones, chloroquinolones, things like your ciprofloxacin and things like that, okay? Which we use for things like anthrax. Okay. So let's talk about cell wall synthesis. So what we're going to deal with is that the cell wall bacteria contains peptidoglycan. Remember, peptidoglycan gets cross-linked. And what that does is make it tough, okay? Makes it tough, makes it durable, makes it hardened, okay? Now, what we're going to do is, since it's tough and hard on the outside like it has a shell, okay? Let's say I wanted to make the shell weak. What I could do is mess up the synthesis of peptidoglycan so that it messes up that cell wall and it gets weak and it will lyse or pop. Now, one group we have what's called penicillin. There's actually about 50 related penicillins, but they all have this thing called a beta-lactam ring. It's this square ring. We sometimes call that the nucleus of a beta-lactam, and then there will be other stuff stuck, but it is this organic ring, square ring called a beta-lactam. Now, we have our natural penicillin that we got from the penicillium fungus, Very narrow, but very good. It's really good for some of your staphylococci and still used for some of your streptococci. It can even get spirochetes like syphilis. But there's a problem. Is your penicillins are very sensitive to an enzyme that some bacteria, like some of your gram-negatives, make, called beta-lactamase. That's how I always called it, but that's how I learned it, sometimes called penicillinase. Now, when you're hearing this ACE, you know it's an enzyme. It's going to basically take this nucleus, this beta-lactam, and crush it, cut it, destroy it, get rid of it. And that makes the antibiotic useless. It destroys it. So it cleaves the beta-lactam ring, and now the antibiotic is neutered. It isn't effective. And so some microbes produce enzymes that destroy the antibiotic, making the antibiotic ineffective. Now what we also did is, to get around this, we went into a lab, figured out the structure that was integral into getting the antibiotic property. And we go in the lab and synthesize it from, like, hey, it's like we have like the natural penicillin is kind of like a blueprint, if you will, to follow from. And it's kind of like, hey, you go to a cookout. The friend makes a side. You're like, that's amazing. And they're like, well, I won't tell you. And you're like, well, it seems like this is mac and cheese with some peppers in it, and you go search, the next thing you know, you find out it's just Jimmy Buffett's macaroni and cheese. You're like, ah! So then you found, because you did something, you're like, oh, you made your version of it. And you make it, okay? Semi-synthetic makes it resistant to penicillinase. So what we did is we get a broader spectrum. Now we can affect more bacteria with it. So we went in the lab, we rattled our beakers, and did better on it. Now, so natural penicillins versus a semi-synthetic. They're going to have that beta-lactam nucleus. And this common bit they all have. What we did is, it's this bit here too. So we modified this side bit. Now, some of these get shot in with a needle, like penicillin G, when you get a shot of penicillin, or penicillin V that we take as a pill. Sometimes we have to give these things like oxacillin and ampicillin. These are resistant to penicillinases, so to get a little bit better, we can extend the spectrum by modifying structure a little bit. Form fits function, guys. Okay, that's a big theme in biology. If you took Gen Bio, if you had me, for sure, form fits function. Structure determines function. So penicillins, cephalosporins, monobactams, carbapenems, they all have that common ring called the beta-lactam. Okay? Now, penicillinase is an enzyme that goes in and breaks down this into something that is not penicillin. It might be penicillic acid. And this is now no longer. Penicillinase is produced by bacteria, especially Staphylococcus. And it makes them really resistant to your penicillins. So there's a few ways we get around this. So what we did is we took our natural, like penicillin G, the prototype, the OG penicillin from the mold, from the fungus. Very narrow, Staphylococcus, Streptococcus, some spirochetes like syphilis. We give a shot, and you keep it about 3 to 4 hours. Then what we did is we took something like procaine and penicillin G, and we call it procaine penicillin. We keep it for about 24 hours. Benzathine penicillin, you basically take benzathine and penicillin G, just up to 4 months, even longer. So you can see what we started to do is take this, what was kind of good, and improved on it. Penicillin V, oral, it's even stable in stomach acid, so it doesn't get destroyed. It's very narrow spectrum, and it's susceptible to beta-lactamase. So these older natural penicillins had big limitations. So there were things it couldn't cover, and we wanted to get more things we could cover. And scientists kind of figured out structure. They found others. They were looking. They still are looking. Now, staphylococcal bacteria started to immediately get resistance to beta-lactams because beta-lactamase. This may have evolved and done so quickly. So methicillin resistance arose very quickly, and we got MRSA. So we don't even use methicillin. It's just, okay, we might as well be just throwing fairy dust around. Then we started to extend the spectrum of penicillins because the natural is really limited. So we want to broaden it. So we also, we can broaden it to get it to some gram-negatives, though they're not resistant to beta-lactamase. So what we did is we got the aminopenicillins. That was our very first extended penicillin. We have the carboxypenicillins that can fight Pseudomonas aeruginosa, very important for people who have cystic fibrosis. It starts to get against some gram-negatives. This is things like carbenicillin and ticarcillin. We had our uridopenicillins. It's basically, we take ampicillin, kind of alter it a little bit, azacillin, meslocillin, sorry. Sometimes I will say these wrong because I'm not used to saying them. It's been a while since patho. Amoxicillin is really big used in kids. And what's really fascinating about amoxicillin, amox, we might give this in a liquid suspension, give this in a liquid, that pink liquid that they take, and they might put the bubblegum flavor in. And they might add that in the pharmacy. What we do with amoxicillin, especially because it's well tolerated by children, safer, we know a lot about it, is that we add this stuff called clavulanic acid. Clavulanic acid comes from a bacteria called Streptomyces clavularis. Now, clavularis is producing this clavulanic acid. It is a beta-lactamase inhibitor. It's a non-competitive inhibitor. We put it with amoxicillin. So when you take amoxicillin and clavulanic acid, we have augmentin, because we have augmented amox. We've helped amoxicillin. So this augmentin is a combination to really get us up to the higher levels of spectrum. Now, we get into carbapenems. Okay, this is another beta-lactam. One of the big things is a carbon replaces the sulfur, so you're not going to have that sulfury bit in here in this beta-lactam ring. They're very broad, much broad. This contains the most broad, one of the broadest spectrum is meropenem. We have primixin, doripenem, and meropenem. A lot of these are penems. Carbapenem, penems. A lot of these are penems. Well, your carbapenems are penems, except for this primaxin. Now, primaxin is a combination drug of imipenem and celastin. Doripenem is really good for Pseudomonas. Again, very useful for the treatment of people with cystic fibrosis who get Pseudomonas aeruginosa infections. Quite easy. Meropenem, your biggest. You can't just give meropenem for everything. It's intravenously. It actually kills bacteria, again, by cell wall, because it's what they do. Really a big one is meropenem for the hospitals. You really want to do sensitivity studies. Make sure you're picking. Is antibiotic stewardship is very important in a hospital. Being careful. And that is something infectious disease is going to get worked up about, is you just like, oh gosh, they just got meropenem for everything. You can't do that. Don't do that. Monobactams, they avoid the penicillinase, a little bit different. They do have this thing here. It doesn't have two rings. It's one ring instead of a double ring, so we call it a monobactam. Is astrenam. It's low toxicity, quite a good. It's good for Pseudomonas and E. coli treatment, who are both gram-negatives. So usually these beta-lactams are not really good at gram-negatives, but these are two common gram-negative infections that we can really affect. with these. Cephalosporins, again, if you have a penicillin-resistant strain, we can use this. They are now really widely used as your cephalosporin, more than any other beta-lactam now, cephalosporins. We have different generations up to the 5th generation going up. I mean, you had the really old ones all the way to the newer ones. And you're getting better even against things like MRSA and the gram-negatives. What we're doing is getting more and more broad or more capable of fighting the things that the bacteria are capable of doing and getting around. Bacteria are sneaky, and they try to get around. Now, bacitracin. We had bacillus that was found in a wound of a patient named Tracy, so we call it bacitracin. It's actually what you have in Neosporin, some of those ointments, antibacterial topical ointment. We can get it from like a staphylococcus infection, a streptococcal infection that might get in your skin. Vanc is our last line against a lot of antibiotic resistance. It is what we call ototoxic, but we give it as an IV. So it damages your inner ear, can lead to damage to inner ear hearing balance, and lead to kidney issues. We want to be really careful with Vanc. Now, if we look at other bacteria that have cell walls, if you look at like a mycobacteria, they have mycolic acids. Mycobacteria, mycolic acids, those waxy molecules. Remember, they're going to get that acid fast name. So what we want to do is inhibit mycolic acid synthesis. Like Mycobacterium tuberculosis. Of course, we want to use something like isoniazid. Ethambutol can again make us not put mycolic acid in the cell wall and be a useful inhibitor of cell wall synthesis as well for things that have mycolic acids. All right, now we can mess up protein synthesis. How can we do this? We can use 70S ribosomes. Now, 70S ribosomes have a 50S subunit and a 30S subunit. Now, what we do to affect this is things like chlorophenol is going to go to the 50S subunit. Your aminoglycosides like streptomycin, they're going to change the 30S bit. So what will happen is, you just with aminoglycosides, you mess up the 30S ribosome bit, and you mess up how we read our mRNA, and we don't make our proteins right. Tetracyclines, these can actually make sure that tRNA doesn't bind to mRNA properly during translation. So we mess up protein synthesis. We don't make amino acids, things like doxycycline. We can go into prevent the 70S ribosomes at the 50S. Instead of 30S, instead of things binding to the 50S and 30S, but the oxazolidiones, I have a hard time with that one. That's not one I say a lot. My girlfriend always giving me a hard time for saying some of these wrongs because she's a pharmacy person. Okay. So we can go in and mess up tRNA binding to your mRNA, or mess up the ribosomes with things like tetracyclines, streptomycins, and chlorophenol, and really mess up this. But we can do it with the 70S ribosomes because they're prokaryotic only, and really gives us a magic bullet, leaving my cells alone and affecting only bacteria. All right, and that is the cool thing. Now, there's some other really interesting things that happen. Nucleic acid synthesis can be messed up. If I want to fight tuberculosis, I can use erythromycin. If I want to fight things like, now, gyrase, that's kind of like a bacterial version of topoisomerases that prevent replications overwinding, so we don't get a replication fork. Ciprofloxacin, gemafloxacin, and moxifloxacin. We can damage our cell membrane with polymyxin B to we put holes in the cell membrane, or we can just put fatty acids in the cell membrane as well, like what we do with treating tuberculosis with isoniazid. Now, one that I find really cool is, besides your sulfonamides and your TMP, SMZ, is they affect enzymes that convert like a nutrient. Let's say this chemical right here, para-aminobenzoic acid. Let's say that's in the media that the bacteria is growing on. And there's an enzyme that turns that into dihydrofolic acid, which is needed ultimately to make folic acid, okay, which is needed to make DNA molecules, okay? So if we go in and we block this enzyme, we don't make this. We can't make DNA. We shut it down. So sulfonamides are going to say, hey, you can't make your DNA. Sorry. Trimethoprim is going to do the same thing. It just works on different enzymes. Now, the TMP SMZ, the trimethoprim, and the sulfamethoxazole prohibits you from making basically dihydrofolic acid. Bactrim is when you take these two drugs together, we call it Bactrim. Bactrim is definitely used in antibiotic. PABA is an enzyme. I don't go into them, but I really want you guys to know, hey, I can't make folic acid if I use sulfonamides. This means I can't make DNA, I can't make RNA, so they're bacteriostatic. They can prevent the growth and spread by the bacteria that are there aren't, that are making you sick aren't killed, but you're definitely not going to get any new ones. Okay, so we can try to treat it with other things too. Now, antifungals quite a bit different. I talk about polyenes who really just are fungicidal. They go to the sterols that are in the plasma membrane of a fungus, and they really just mess up and kill the fungus. There's azoles and allylamines. They mess with sterols as well. So this is like, hey, for like jock itch, ringworm, athlete's foot, and so-called yeast infections. They're found in a lot of the lotions that you put on, a lot of the creams, the anti-itch creams that are used to treat things. Ringworm is not a worm. It is a fungal infection. Jock itch you get in your crotch, athlete's foot on your feet. Yeast infections can be found in different places, but oftentimes reproductively. We have yeast infections. When things like you go on antibiotics, you may get a yeast infection, your pH balance gets off, things like that. Echinocandins, they basically lyse the cell wall of a bacteria, or the fungus, sorry, I said bacteria. Bacteria do have cell wall, fungi have cell walls too. One of these, and I have the hardest time saying this one for treating ringworm skin infections, is greasofulvin. And I may have not said it completely right. I'm not by any means an antifungal expert. I know a lot more about antibacterial, more experience and expertise in that. Was I taught this stuff? Yes. Is that what I spent some of my time? No. Now, antiviral drugs. Basically, viruses aren't really alive. They're alive when they're in you. They need your stuff to do things. They can't replicate. They don't have metabolism. They need a cell of yours to get in. So they're very hard to target because, like, hey, you can hurt your own cells. They have a high mutation rate. They tend to mutate when they infect, when they go from one host to the next host. That's kind of when they really have the mutation. They're not so much as inside you, but when they go from host to host, they have some mutations. Interferon is definitely used to treat viruses. There's different kinds of interferon, but alpha interferon is the big one that prevents cells. If a virus infects a cell, that cell makes alpha interferon to warn the other cells that the virus is coming. So inhibits spread. So HIV, we're going to talk about this. There's an enzyme HIV and other retroviruses have called reverse transcriptase. And what reverse transcriptase is going to do is you're going, transcribe means you turn DNA into RNA. Well, you're going to take RNA and turn it into DNA. And then you're going to integrate that DNA into the patient. So we use what's called reverse transcriptase and integrase, and we'll talk about those enzymes later on in the viral chapter. Antiretrovirals, that's what we use to treat HIV and some other retroviruses. Retroviruses scare the heck out of me, but basically you prevent that replication. If you can't make RNA from DNA, because basically you're going to take a reverse transcribe to turn their viral RNA into DNA and integrate that DNA into your cells. Protozoans can be pretty hard to fight. Usually it's anaerobic metabolism. Here we have a nice little, I do believe that is a Giardia. Giardia lamblia, yes, that is a Giardia. Little Giardia, which causes, I like to call it beaver fever, Giardia lamblia. Quinine was one way we used to treat a protozoan, malaria. There's a plasmodium that does that. Now we use chloroquine. Quinacrine is we use that to treat giardiasis, which is caused by Giardia lamblia, which is you get that in, like, you might drink from a creek, and I like to call it beaver fever. Not beaver fever. Beaver fever is a disease in which there is no cure for. Antihelminthics, a nice little look up at the scolex of a tapeworm there. So helminths, parasitic worms, tapeworms, we can use niclosamide to kill tapeworms by starving to death. We don't make ATP. Parzaquantal, I do have to really be careful saying these, just makes the cell membranes of tapeworms become permeable and they die. Mebendazole. There we go. Mebendazole. It makes me think of trying to think what it made me think of. Anyway, you can't make microtubules, so they really have problems with things like cell division. They also don't absorb nutrients. Streptomyces actually makes ivermectin. Ivermectin is what we call a horse dewormer. It is against worms, mites even. We don't exactly know. We know it paralyzes and kills the helminth. We don't exactly know what ivermectin does. It is a livestock treatment. As I said, a horse dewormer is kind of what it's really known for, is ivermectin. Antimicrobial drugs. Now, this gets really interesting, and I really want to talk about this. I really like talking about is that when you have antibiotics, you start to have an evolutionary arms race. It is like a weapon. And I kind of like, okay, back when I was young, the Cold War between us and Soviet Russia was still going on. Soviet Union and America were in a Cold War. We still have the fear of nuclear annihilation. And basically, kind of like an evolutionary arms race, okay, we sometimes call this Red Queen hypothesis. Remember the Red Queen in Alice in Wonderland? She said it takes all the running you can do to stay in the same place when they were playing croquet on the ever-changing landscape. Well, this is kind of what happens, okay? Antibiotics, kind of think of that as a disease for the bacteria. And what do I mean by that? It is a danger. It is a harm to that bacteria. Now, bacteria aren't going to just sit around and take it without fighting back. Now, what happens? Let's say that I've got affected with an antibiotic, okay? And a bacteria, sorry. I get infected with a bacteria. And I take antibiotics. When I first get that, the bacteria are very susceptible. There's a high mortality rate, and we wipe out. We almost decimate the bacteria. And there's only a few survivors. Now, what we call this is a genetic bottleneck. The survivors have some genetic characteristics that let them survive for some reason. Something in their genetics gave them an advantage, and they survived. And when they reproduce, their offspring, their progeny have that resistance. And now they will grow. Random mutations can create this. One of the things I used to do in a lab is we would use random mutagenesis processes like bombarding bacteria with UV light. And UV radiation causes anytime two thymines are side by side on the same strand, they bind side by side, not across. And then the across bit ends up becoming mutated, and you get what's called thymine-thymine dimers. This is how it actually causes skin cancer, and actually how it can cause mutations. And the thing is, we would then take the bacteria that we bombarded. Now, this bacteria was not antibiotic resistant. We could bombard trillions of cells, plate them, and we would put them on a plate that had antibiotic in it. If we did enough of this, we would get an antibiotic-resistant bacteria from our experiment. Then we could see how they got it. What is it that gave them the antibiotic ability? So we would cause it, and then we could study why. What happened? What is it that happened genetically? So random mutations can generate an antibiotic resistant. There are drug resistances on plasmids, extra bit of DNA, like what's called the R factors, bacterial conjugation. They share it through bacterial sex. Now, I want to show you this because this is frightening to me. I'm going to play this video. Very frightening.
So we ended up building them basically except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands. At the base of each of these bands, we put a normal traditional agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, of course, in agar, that bacteria move around it. The background is black. I think bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it.
And these mutants all have different ways of being resistant for different reasons, different random mutations.
See the different mutants. Repeat this at 100. And after about 11 days, they finally make it.
11 days it took. Now, what I want to tell you with that, now, if you want to watch that video all the way, but that's really all I want to show here. All right. Now, how can they get this? Well, how do they get resistant? One thing is they can just say, hey, antibiotic, you can't even enter. We can prevent the antibiotic from going in. They can close their portin proteins in their cell walls. Be like, oh, sorry, I'm not going to let it in. And it's kind of like, hey, you know there's danger, so you lock your doors. You shut your windows, lock your windows. You grab your gun, and you ready to defend yourself. Okay? We can deactivate or destroy the drug. We can actually break down, like what happened with bacteria having a penicillinase or a beta-lactamase. We can take the molecule, like that, where the drug targets. We can change that. Now the drug can't bind to it. Or we could just pump the antibiotic out, like it was a toxin. They have pumps to get rid of toxins, and they're like, hey, we'll just use that pump. We'll pump the antibiotic out. It's like, well, I got the antibiotic, but I removed it. It didn't affect me. I am scot-free. So these are all ways that antibiotics, that bacteria can get around this. Remember, this is like a war for them. This antibiotic is an attack on them like it was a disease to us. And they are fighting it. And they will quickly come up with ways around it because life finds a way. And it keeps going. Okay? This is how this works. Very intense. And what can happen is antibiotic resistance will come about pretty quickly after you start giving the the antibiotics. And we get what we call a bottleneck. So basically imagine I have this population of, some of these are just, like very, 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 very susceptible to the antibiotic. Many of them are middly, and then a very few of them might be really resistant. And we have this wide varied population. The antibiotics are taken, it wipes out a bunch, leaves behind the resistance, now you have antibiotic resistance. Now you have a resistant bacteria. How do we prevent this? Is misuse is a big thing, okay? Using an outdated antibiotic, weakened antibiotic. People will do things like, oh, well, I've got this antibiotic when I have my tooth taken out, I've got it. Don't do that. Do not do that. Take antibiotics that are in date and are given by your doctor at the time, and take all of it. Like, oh, I've got a cold, I'm just going to take an antibiotic for a head cold. It's not going to help you. Antibiotics in animal feed, they do that widespread in factory farming. Not taking the whole thing. If they give you a whole bottle, you take all of those pills in that bottle before you are stopped. Do not have leftover antibiotics. Now, somebody's like, well, I had my tooth taken out last year and I have some antibiotic pills. What you're doing is they're leftover pills. That is contributing to antibiotic resistance because you don't have enough there to fight the infection. Take what the doctor gives you, all of it. Take it as prescribed. Do not leave a pill. Take the entire bottle. It is a fight you see to the end. Okay? It is, remember, what doesn't kill you will mutate and try again. Okay? All right. So one of the things I find really cool is this idea in biology called synergism. And there's sometimes it's like, hey, one antibiotic don't work, and the other antibiotic don't work. Put them together and bam. 2 drugs together are better than one by itself. We call that synergism, working together synergistically. Antagonism, okay, you have competing. I'm making it worse. It's less than, because I took these drugs are antagonistic. They make it worse. Okay, they lower the efficacy by taking this with it. Okay, so there are things you can't take with antibiotics because they make antibiotics not work right. Okay. One thing I was involved with in some of my research at some point in my education was looking for future chemotherapeutic agents. I was involved in working with defense peptides that were found in the skin of salamanders. But we'll look at these antimicrobial peptides like found in plants or animals. So I was looking for one that was found in an antimicrobial peptide in the skin of a salamander. It actually made ATP synthase in a bacterial cell run backwards. And there's another one from an amphibian skin gland called magainin. Niacin, it's a lactic acid bacteria that makes an agent. I have never actually said that a silk moth makes one called sacropin. Melittin is actually from a bee venom. I don't know what species of bee. There's many defensins that are made by white blood cells like neutrophils that many animals and plants make. Bugs, plants, birds, mammals, they make some of these defensive molecules in the white blood cells that become antibiotic, chemotherapeutic that we can look at. So there's lots of places. We are looking at the new biotics, the antisense agents that are DNA strands that actually block bacterial toxin production. So the bacteria is there just as neutered. The bacteria can't produce a toxin, can't make you sick. One of the things we're also looking at is how do they infect us? We want to try to prevent that. There's RNA interferences. We have what's called siRNAs. This can help us with virally infected cells, stopping gene expression. There's what we call small interfering RNAs. You can't make proteins. They bind to an mRNA. It's double-stranded. Now you can't read it. You can't read the mRNA. Spread viruses like retroviruses can't spread. Small interfering RNAs mess that up. They mess that up. They go in and they now make basically this RNA a double-stranded molecule, and it's not right. All right, that's it, guys. I went ahead and re-recorded that because there were some things I wanted to do to improve it. I may look at other ways I want to improve in the future, but thank you, guys. I really hope you enjoyed it. I wanted to give some passion as well and things that I was involved with as well to kind of tell you a little bit more about me as a scientist. Thank you, guys, and I can't wait to hear from you. Thank you. Have a great day.